All right, thanks everyone. Really good to see so many people attending today. As Jackie said, you'll be able to look at this on the YouTube channel later on, but this is part of a really big project that's been running over the last 18 months. It's been a collaborative project between EnviroDNA and Cesar Australia and funded through the Australian government. There's also a number of other collaborators and people that were involved along the way that I'll acknowledge at the end. But so the overarching goal for this program was to understand whether those devastating fires that happened over the 2019-20 summer had an impact on platypus populations. So platypus were identified as a priority species following those fires, and some real concerns about what the impact might have been. On the back of, I guess, a lot of concern recently about the status of platypus populations, how they're going, what are their sort of main threats. So I'll just back up a bit and I'll start with some of the major threats that we know that they face. So one of the challenges we have in studying platypus populations is that we really don't have great data about how populations are going, particularly historical data to be able to compare against, which makes a real challenge in being able to understand whether there's been declines or not. But I guess the concern stems from platypuses are dependent on aquatic ecosystems, of course, and we know that a lot of our waterways throughout Australia are quite modified and degraded. So some of the major threats that platypus face are things like a reduction in available water through things like drought, and we know that there were some pretty significant declines, at least through the southeast area of Australia during the millennium drought. So what that does, it just reduces the amount of habitat that's available for them. It fragments populations, it reduces their food supply, impacts water quality, vegetation. Not surprisingly, drought isn't good for an aquatic species. Exacerbating that is the fact that we need water for our own uses. So we extract water, we divert it, so it puts platypuses in competition with us for a finite resource. As part of that, we also, sorry, I just saw a hand raised there, Jackie. I just want to make sure that everyone's hearing me properly.
Check.
Perhaps if there's any issues, you could pop it into the chat function.
Yeah.
But yeah, hopefully people are able to see my slides and hear me talk. So yeah, things like dams, again, diversion of water, change flow regimes up and downstream, and importantly are a physical barrier for dispersal, so they can fragment populations. And then outside of the water, what goes on in the surrounding catchments are really important. And of course, a lot of our catchments are highly modified, either through urbanisation or agriculture or industry. And so clearing that vegetation around waterways has a significant impact, as well as in the broader catchment area. We have invasive species, we have woody weeds that take over our riparian zones. And then things are compounded by having things like stock access that further erode banks and potential to introduce pathogens. So we know that there's a lot of threats that platypus potentially face. We don't really have a good handle on the extent or the severity of some of these threats, which are things that hopefully we're starting to learn more about at the moment. But I guess the key thing is fires haven't really been considered a threat to platypuses before. But when we think about what their key requirements are, so platypus really require three fundamental things in their habitat. First of all is water, obviously being an aquatic species, and in with that is flow regimes and water quality as well. But I guess what's most relevant for this project is they require banks for burrowing, and as part of that is stabilising vegetation on those banks, which holds that soil together and enables those burrows to be constructed. And I guess most importantly, they need food, they need macroinvertebrates. So and they eat a lot of it. They're not really fussy about what they eat, but they do require a fair amount of food. And a lot of the habitat variables that we know are important for platypus are really about supporting abundant macroinvertebrates. So, and that's what we think is going to be impacted by fires. So I guess one of the reasons that platypus, or I guess bushfires haven't been considered a major threat before is that being an aquatic species, they're somewhat buffered from those direct impacts. So, you know, we all saw those horrible photos of koalas and kangaroos that were burnt by the fires. Those direct impacts are not really what impacts platypuses. So they're going to avoid that. It's what happens after the fires that we think is important. And so when we look at a burnt landscape like this, you can see that all of that vegetation, all that ground level vegetation is gone. So if we get rainfall following those fires, all that loose soil and sediment is going to wash into the waterways. And of course, there's a heap of ash mixed in there as well. So all of that inputting into waterways is going to impact their food supply. It's going to impact stability of banks. And I visited a few areas not long after the fires. It was about February 2020, and saw situations like this where there was a lot of sort of black debris in our waterways and this sort of ashy sludge on the benthos. So, and we know following fires, you can get things called black water events, which is when you get these big slugs of this sediment traveling down a waterway, results in really low dissolved oxygen. You can have mass fish deaths. And there's variable impacts on macroinvertebrates. And so clearly, if we're going to reduce food supply for platypuses, that's going to have flow-on effects. So while they don't have those direct impacts, there's going to be longer term effects over months or even years around body condition and survival and juvenile recruitment. So it tends to be more subtle impacts that we expect, and I guess that's why it makes it difficult to really understand those impacts. And where the few studies that have actually looked at fires on platypuses have been really inconclusive, and a lot of that is because there's been they've really been hampered by not having any pre-fire data to compare against, or not having adequate controls to compare, you know, in unburnt areas. And hopefully during this project, I'll show you the importance of having both of those sets of data to compare against. Now, I'm sure a lot of people have sort of seen this. This is a map of the main fire extent, and seen the, you know, some of the statistics around that. So it's important to recognise this wasn't a single fire. It was a whole series of fires over about 3 or 4 months. These fires were unprecedented in terms of the scale that they impacted, and also the fire severity. You know, it was estimated to be over 11 million hectares and 3 billion vertebrate animals that were impacted. And I guess really importantly for this project is that a lot of these areas overlap with known platypus habitat. And importantly, it's what's been considered really good platypus habitat. So in Victoria and southern New South Wales, it's those sort of relatively undisturbed forest areas. And while we haven't had great data on populations, we consider that to be really good platypus habitat. Same as up in northern New South Wales here and the Blue Mountains. So a lot of those sort of forested areas that are really good habitat for platypus and a lot of other creatures are the areas that were most significantly impacted. And University of New South Wales estimated that it was 13.5% of the total available habitat for platypus across Australia that was burnt. So even if the fires have a subtle impact, the sheer scale of them is going to have serious consequences for platypuses. And I guess to understand the impacts of something at this scale, you really need to generate data over a large scale. And we simply haven't been able to do that previously. So it's important that we're not just looking at a couple of different waterways or a small area. We really need to look at things over a large scale to understand large scale impacts. And also platypuses are incredibly widespread and dispersed. So it's really hard to extrapolate from quite localized studies over to a much larger area. So the reason we're able to do that now is through the evolution of a monitoring technique called environmental DNA. I'm sure a lot of you are familiar with it, but for those that aren't, just to give you a bit of background, it's based on the premise that everything is shedding DNA into its environment, and now our genetic techniques are powerful enough to detect these tiny little traces in things like water samples and soil samples, and even more recently getting pulled out of the air, which is pretty exciting. Some of the advantages of this is that it's not invasive. It means we don't have to see or trap or disturb the animals in question. It means there's no animal ethics issues. It can be really cost effective to do, particularly over large scales like this project. The sampling methodology is really simple. Again, importance for this project is that we've had a lot of collaborators collecting samples or doing their own projects and contributing data, including community groups. And I've even had, school groups doing this before as well. Now, for any monitoring technique, the key thing is it actually effective at detecting your species? And we know that eDNA is highly sensitive. Just to give you a little bit of data on that. So this was a paper that was published a few years ago, just demonstrating that. So this was in the Melbourne region in some sort of small to moderate sized waterways. And we were showing that with, a couple of water samples and a couple of replicates in the lab, we can get over that magical 95% detection rate. So we know it can be really effective at detecting platypus, even at low densities, which is important. And conversely, if we're not detecting platypus, we can have pretty high confidence that they're not present. Now, there are some things that impact that, which I'm going to get to a little bit later as well. So with this technique, it means we can do things over large scales that we haven't been able to do before. We're not as limited by conditions or access to areas or where people are available. And I guess importantly for any project, it means you can design a really rigorous sampling program that's going to answer your question. So, and again, hopefully I'll highlight the importance of having a good survey design in this project. So this was our area of interest. This grant was very much focused on New South Wales. It was the state that had the biggest area impacted by platypuses. We did incorporate some data in southern New South Wales, southern Queensland, up in this region, from some collaborators who are doing a very similar project up there. They kindly donated that data to this project, and it made sense given that platypus and fires do not respect state boundaries. So it enabled us to increase the scope of the project as well as sample size for analyses. So when we were first doing this project, what we really wanted to do was try to do a before and after comparison. Now, because no one was able to predict these fires, there was no sort of systematic pre-fire data collected across that area. So we were reliant on records from wildlife databases. So these are the red markers here. When we started looking into it, we discovered that, probably not surprisingly, they were pretty sparse. And typically with these anecdotal records, they're clustered around population centres. And so a lot of these fire affected areas, which were in more remote national park areas, had very few records. That doesn't mean platypus weren't there. It's just that they have very few people there looking for them. And so that really made it difficult to do a before and after comparison. So what we did do, we expanded that data out to 30 years, which is the yellow dots there. So 30 years is approximately 3 generation lengths for platypuses. And that's the length of time that IUCN used for conservation assessment. So that's why we chose that period. And basically for each of the subcatchments that we wanted to look at, we only looked at areas where we know platypus had previous records before. So we wanted to make sure we were looking in areas that we know platypus were present, rather than areas where we had no idea that they were there before. What we did have, which was really valuable, was, when I say there was no systematic data collected, there was in the southern part of the state. This was a PhD project by one of our people that we worked with, Emily McCall Garsden from Melbourne University, who'd done some surveys through Victoria and New South Wales. And so we actually had a series of eDNA surveys in the southern part of the state that included pre-fire data. So that was a really valuable data set that we used as a little subset for analysis, which I'll get to in a minute. But it meant that we actually had some good and rigorous pre-fire data to compare with. The project was also, well, there were 4 main regions that were identified through the grant area, and they sort of correlate with the main fire affected areas. So we had South Coast, Alpine, Blue Mountains and Northern Rivers. And then, as I mentioned, we included some Southern Queensland areas as well. And so our survey design was distributed across those 5 main regions, and included both burnt areas as well as unburnt areas as our control. And again, that was really critical for the outputs. Of course, there were a few challenges, as there always are. I think the start of this project was actually still impacted by COVID. I think the New South Wales border may have still been shut initially. But I guess the overarching challenge for the last 12 months, for those of you that are in New South Wales, you'll be probably painfully aware of this, but there was a few storms that came through. And so there was mass flooding, and even just elevated river levels that were, well above normal, impacted our ability to get to sites, but also impacted suitable conditions for eDNA sampling. So if you've got a lot more water around, then intuitively it's going to dilute your DNA that's in the water, and so your detectability is going to be reduced. The other thing that it does is that when there's heavy rainfall, it washes a lot of sediment and debris into the water, which clogs up your filters when you're doing the sampling, and it means your sample volume is less. So it's sort of a vicious feedback loop. Despite all of that, we did manage to get out there with help from a lot of different groups. So we had different people doing sampling for us. We were able to share samples with a few different groups that were doing other types of projects that they then allowed us to screen their samples for platypus. And then there were some key collaborators who were doing their own platypus projects that donated data as well. Oops, jumping the gun. So as I mentioned, we were looking at this statewide assessment, and I guess the key part of our project design was looking at both impacted areas, so fire impacted areas, as well as unburnt control sites. And so as part of this project, we did, it was just over 400 sites that we went out and sampled. And so here we've got, so our green dots here are where we detected platypus. Our grey dots are where we didn't detect them. And then the yellows are what we're calling sort of possible detections. You find very faint traces of DNA in those samples, which quite possibly indicate platypus presence at low density. But they can also come through contamination. And so we don't confirm them as platypus presence. We leave them as this sort of possible detection. And then, as I mentioned, we had a number of collaborators that donated some data. So we had National Parks and Wildlife Service that did a project through the Blue Mountains area. The Jalagir Biodiversity Alliance did a couple of projects up around the Coffs Harbour, so we got some high density sampling there. We also had Blue Mountains Council, again through that Blue Mountains region, and then New South Wales Department of Environment in the Alpine region. So all up, we got really good coverage across the state. And then we also had our samples from Wildlife Queensland in that Southern Queensland region. So really good coverage throughout those fire affected areas, those 5 main regions that we were looking at. We ended up with, well, we well exceeded what we were expecting to do for the project. We ended up with over 700 sites that we got to, which enabled us to do some really, really good analysis. And then this data was used by Emily McColl, so at Monash Uni now, to do some site occupancy modelling using those data sets. And there were 2 main data sets that we used. As I mentioned, we had the selection of sites in that southern New South Wales region where we had pre-fire and then a series of post-fire sampling over the last couple of years. And then we had our broader data set where we didn't have pre-fire data, so it was a comparison between our control and impact sites. So we'll start with our subset in southern New South Wales. So a lot of this is part of Emily's PhD. Her thesis is finished, and hopefully this fire paper is soon to be submitted and published. So these first 3 time periods here are from Emily's data. So orange is our burnt sites, and green are our unburnt control sites. So initially, pre-fire, we had slightly higher occupancy in our burnt areas. So this is reflecting most likely better habitat. So a lot of those burnt areas were that sort of nice forested areas or native veg areas, whereas our control sites, as much as we tried to keep them similar, incorporated some more agricultural modified landscapes. So the most interesting part here is what happens post-fire. So we saw a slight decline in occupancy in our fire-affected areas in both 2020 and then early 2021. Over that same time period, we actually saw a slight increase in our control sites. And then from this project, our 2022 samples, we saw a pretty significant decline in both of those groups. So, as I mentioned, having a good survey design is really critical. And without our control sites here, we might have concluded that we've got a delayed significant impact of fire. But because we're seeing a decline in our controls as well, it's most likely reflecting the habitat conditions during that 2022 period, and in particularly those elevated flows and storm areas. So we suspect that this is probably impacting both platypus occurrence. So we're probably getting displacement of animals from those high flows, but also slightly reducing our ability to detect them through eDNA. Now, when we look at our different variables, I won't get too detailed here, but basically anything to the right of this central line is a positive impact on occupancy, and anything to the left is a negative impact. Where these confidence intervals cross that center line, it's a non-significant effect. And what we saw here was our occupancy in our fire-affected areas was higher. Bit counterintuitive, but it's largely driven by this initial pre-fire occupancy. As we can see, post-fire, it's actually starting to drop below our control sites. But the interesting thing for me is this interaction here between fire and our time period. So post-fire, we saw negative, a reduction in occupancy in all 3 time periods, really high variability here, which makes it really difficult to make them significant, but it's showing that there has been a slight decline in those fire impacted areas compared to our control areas. Now, when we look at that much broader data set, so this is our statewide data. So we had 631 sites here. There was 100 of these that were, or around 100 that were visited on multiple occasions, which brought us up to our 700 odd sites. Now, our occupancy and our burnt versus control areas are really similar here. And so, again, looking at good survey design, if we were to take this in isolation, we would just say, look, there really hasn't been any impact of fires, but we know from our pre-fire data that our occupancy was actually higher in those fire-affected areas and has subsequently declined. So, again, really important that we've got that strong survey design. So, but I guess on the plus side it's also showing that there hasn't been sort of major impacts of those fires. So both our burnt and unburnt are having pretty similar occupancy rates in 2022. Now, for this study, we also incorporated a land use variable. Now, we simplified it into just 2 variables here. So what we're calling forest is basically our native vegetation areas, and then we have our modified landscapes, which is a combination of agriculture and urban. The vast majority of our sites were in those forested areas. I think it was around just over 500 were in our forest land use, and about 100, 120 were in our agricultural areas, which makes it hard to show impacts of that land use. But we did have a positive impact of forest, and we know from previous work we tend to get really high occupancy in those areas. It's also what's driving that higher occupancy in our burnt sites pre-fire. But again, the interaction terms are what's interesting here. And I should say we've got a slightly negative impact of our modified landscapes. But these interactions are really interesting here. So the interaction between fire and our forested areas is showing a negative occupancy, and compared to our agricultural areas, which are actually slightly positive. So the impact of fires in forested areas is much more significant than what's in our agricultural areas, which is what we'd expect. Fire severity in those forests is going to be much higher than what's in our cleared landscapes. And just finally, one of the other things that we sort of did as a bit of a add-on to this project, I mean, one of the things that we really don't have a good handle on for platypuses is population declines and the level of declines and the extent of declines in both at a local and also our catchment level. So we use that sort of data set of 700 odd sites to look at how many of these sites do we actually have historical data. So using data from the ALA, we put a buffer around each of our sites. I think I might have, oops. No, I thought I had some info there. We put a buffer of 5 kilometres around each of our survey sites. So that's to incorporate both movement of platypus, movement of DNA, which we know that gets transported with flow, but also a bit of uncertainty around location data from our databases. So we use that 5K buffer, filtered out which sites we've got previous platypus records. So within that 30-year period, how many of these sites did we have records of platypus being there previously? And then looked at, did we detect platypus during our current surveys or not? So the white markers on this map are where we did detect platypus. So these are sites we know platypus occurred there previously and we've still found them. And what's concerning here is our red ones, which are the ones that we didn't detect platypus. Now, I should point out, this is very preliminary analysis. There's a lot of other things that need to be investigated as part of this. There's also the issue of what sites where we don't have previous data, what do we do with that and what's the significance of those? But it is highlighting some real concerns here. What was interesting is that we saw those declines in both our forested and our modified landscape. So I would have thought that we'd see greater declines in our modified landscape, but they were almost identical. But concerningly, it was about 60% of sites that we had previous records of platypus that we didn't find them this time. Now, we know that's going to be impacted a little bit by the conditions during the current survey, but it certainly is highlighting some serious concerns throughout New South Wales, really. It was pretty similar declines across all of the 5 regions that we looked at, a little bit higher in the southern area and a little bit lower in this northern rivers. Again, that probably reflects the habitat quality. We expect this habitat quality in the northern area to be relatively good. But yeah, certainly highlighting some issues across the state that needs to be investigated a bit further. So just to sum up, I guess the whole point of this project was trying to understand, did those fires have an impact on platypuses in New South Wales? And I guess the overarching answer is yes. We were only able to detect that with our subset of data where we had pre-fire data to compare against. And we think that the impact has been relatively minor. It was only a decline of a few percent in occupancy. But the worrying thing is simply that the scale of the impact of those fires. So even a decline of a couple of percent over a very large scale is going to have a significant impact on platypus populations. Now, I do expect that populations are going to bounce back in these areas. They tend to be pretty good habitat areas. The question is how long it takes and whether there's any further disturbances that are going to, compound the problem in the future. What we did see was that things like land use practices probably have a much bigger impact. Our occupancy in our modified landscapes was quite a bit lower. That reflects what we've seen in other programs as well. And again, something that can be investigated a little bit more. And as I mentioned at the end there's some worrying, again, stressing preliminary data on potential declines across New South Wales. I guess some of the really good impacts or one of the key outcomes of this project is that we now have for the first time, a systematic baseline data set on platypuses across New South Wales. So we've now got much greater capacity to look at changes in the future, whether that's from future disturbances or management actions, so that we can see whether they're being effective. One of the things I think is really interesting is what has the impact of flooding been, both on these results, but also on subsequent data. So, what impact has it actually had on platypus populations, which, we're not going to know for some time yet. So I think that's another thing that's worthy of investigating. All right, I'm starting to get a croaky voice, so I'll leave it there and we'll get on to some questions.
Thanks, Josh.
Actually, sorry, Jackie, just before we do, I'm just going to quickly jump ahead. Or else I'll miss this, which is really important. This project was certainly not done in isolation. Lots of different organisations contributed, both in terms of in-kind data that they were able to contribute, collecting samples, donating samples. So a huge thank you to each and every one of these organisations that were involved. I certainly hope I haven't missed anyone off there. If I have, I apologise. But I'll get you in the report. Okay, thank you.
Well, thanks, Josh. So the first question we got is just about the recording, and we are recording this session and it's going to be posted on our YouTube. So I've just posted the EnviroDNA channel link into the chat. And now on to some other questions. So there was a question and a couple of discussions around flooding. And the first one that came through is, does the increased water flow make it less accurate to assume platypus were in that stretch of river and not 30 kilometres away, for example, if it was severely flooding?
In terms of, is it transporting DNA further? Yes, I mean, certainly not to that extent. But so there's two things at play there. The extra water is going to dilute the DNA, so the possibility of detecting it is going to be reduced somewhat, and no one's really got a good handle on how significant that is. But also it's with faster flowing water, it's probably going to transport further, but also it's going to dilute further. So yes, there's a possibility that it travels a little bit further, but it's probably more like a kilometre rather than 30 kilometres, based on everything that's been done around DNA transport previously.
Okay, on to the next one. So where you say burnt sites, were they already burnt at the pre-fire data point, or do you just mean areas that later became burnt by fires?
Yeah, so sorry, these are areas that were burnt during those 2019-20 fires. So none of them were burnt in the pre-fire area. I mean, some of them were probably burnt historically, but no, this is all just, when I'm talking about burnt, it was stuff that was burnt in that 2019-20 area timeframe. We also put a buffer around those the fire extent as well of 10 kilometres to sort of incorporate downstream impacts as well, just to clarify.
Another one is, could some increase in occupancy at non-burnt sites from pre to post fire be because of platypus moving to them from burnt sites soon after the fires?
Yeah, interesting. I have considered that as well. I think mostly so yes, it is a possibility. However, I think most of our control sites were sufficiently distant from the burn region that it would be unlikely to be most of it. I mean, there's going to be some natural variation as well. We know we had pretty decent rainfall during 2020, 2021, which probably helped ameliorate the effect of the fires, but also we know that that rainfall is going to be good for platypuses, so they're going to reproduce more, they're going to increase in distribution and abundance during that, under those conditions. So, but again, really highlights why we need those control data sets for comparison.
Another one is, how mobile are platypuses? What would constitute an insurmountable barrier? Can they walk around a dam wall?
Yes, so platypus are really mobile, both in the water, they'll travel kilometres, so their typical home range is sort of 2 to 5 kilometres. We've recorded a young male travelling 55 kilometres over 9 months, I think it was. So they're very mobile naturally in the water. In terms of getting around barriers, yes, so these barriers are they're much less of a defined barrier than they are for, say, fish, because platypus can leave the water and move around them. I guess the key thing is they don't really want to unless they have to. So when they leave the water, they're very vulnerable to predation. They're very vulnerable to overheating if conditions are hot. So if it's cool conditions, it's highly vegetated, there's no predators, they will move around some of these barriers, at least occasionally. But I guess the key thing is these big structures, and depends on what the size of the structure is, they may not be complete barriers, but if they're stopping 80% or 90% of movements, then that's a problem. So yes, I think anyone that talks about dams being 100% barrier, it's not a yes/no answer. It's a how much of that movement are they stopping, and they're certainly stopping some of them. Smaller dams might only stop 10% of movements, but those large dams are going to stop most of them. And there was actually a paper that came out recently around genetics up and downstream of some of these large dams, showing that there's isolation across those barriers. Having said that, I've seen them traverse massive waterfalls as well. So they can do it. It's just very unlikely.
Someone actually said that they like your shirt, Josh.
Thank you. I'm really disappointed now that it's all online, that no one gets to see my my platypus shirt anymore to know that I'm a platypus ecologist. Thank you.
And another question was, did you look at burn severity classes for your burnt areas, i.e. FESM data?
We didn't in detail. In fact, I was going to mention it, the data that Emily used for her PhD, as well as showing an impact of fire, she went into it in a lot more detail and looked at fire severity and, really interestingly, post-fire rainfall. So I don't want to steal her thunder, but there'll be some really interesting info coming out about that. And certainly, yeah, both of those things have an impact. So greater fire severity, greater impact. And then what happens post-fire is really important as well. But yes, Emily has looked at that. And so keep an eye out for her publications.
And in a similar kind of way, someone has asked, have you looked at delineating the data based on burnt and flood and just burnt?
We haven't, and I'm not sure that there's too many areas that didn't flood. Certainly, you know, those northern rivers were the most impacted, but virtually everywhere in New South Wales at some stage during 2022 had some level of flooding, whether it's minor or major. I think it would be really interesting to try to tease that out, but it won't be part of this project at the moment.
And another one, did you notice much of a difference in sensitivity between samples collected using a syringe versus those which used electronic samplers?
Yeah, no, not noticeably. I mean, it's really hard when they're not direct comparisons. We have done a little bit of that comparison internally, looking at our syringe filters versus our Smith root pumps. So different sampling system as well as slightly different filters that are used. There is a little bit of an impact. So when you're using our Smith root pumps, they filter more water. You do collect more DNA. In terms of practical sense, it doesn't seem to make a huge difference in detectability unless you've got really rare species, low amounts of DNA. You're going to get a little bit higher detection using the Smith root pumps. You filter litres rather than millimetres. Yes, we can incorporate a filtration volume as a covariate in that analysis. But yeah, I think from what we've done previously, there's not a huge difference there.
And how did you track that male which travelled 55 Ks? Are you, are you able to distinguish individual animals with a really good quality eDNA sample?
So no, that wasn't through eDNA. That was through a long-term trapping program that's done in the Melbourne region. Every time we trap one, we microchip it, and we just know from where we trapped them in different areas. We trace the shortest route. So he might have done more than 55 Ks, but if he took the shortest route, that's his minimum distance that he traveled over that time period. And just on the eDNA thing, so no, we can't identify individuals from eDNA yet. Watch this space, I think, because it's developing very quickly. I think it would be an amazing tool to be able to do a mark recapture study with eDNA. That would be phenomenal, but not yet.
And another one is, what time of year is the best for platypus eDNA sampling?
Yeah, it doesn't matter too much for platypus, as opposed to some other species. Certainly things like frogs, it's very seasonal for eDNA. You want to make sure they're in and around the water. For platypus, they do have higher activity during breeding season. So in this southeast region, it's sort of August to October. So we do try to target those areas. And again, it was something that we would have liked to have done for this project. However, I think in the first year, we couldn't get across the border for breeding season. And then with just all the storms in 2022, our field work just ended up being spread out over a much larger area. Having said that, it doesn't make a great deal of difference. The other good time for platypuses is during autumn, when you're getting juvenile emergence. We tend to avoid the summer period just because hopefully a lot of adult females are in the burrow with their young, and so you've only got males that are, well, males and some of the females that are out and active. So intuitively, there would be slightly less DNA around during that summer period.
And we have a slightly different question. Is airDNA monitoring happening already? Great talk.
Yes. In fact, we are doing some R&D in that space at the moment. Yes, it's happened. It's been demonstrated as a proof of concept. It can detect DNA. There's still a lot of R&D left to be able to work out exactly how effective it is, what sort of taxa it detects most effectively, what type of environments it might be good at, what's the best way to sample air. So it's very much in its infancy, but shows a bit of promise. So yeah, watch this space and we might be able to answer some of those questions shortly.
Doug's asked a question. He's talking about an article that was published in November that spoke about how large dams may also threaten the survival of platypus populations. How big of a threat do you think dams are, say, versus fires and other threats you mentioned?
Yeah, so that paper was really around dams as barriers, so fragmenting populations. I think the biggest impact of dams isn't so much the fragmentation, it's what it does to waterways. So changing those flow regimes, diverting water. Now we can manage that better. So we've got a much better understanding now about environmental flows. We have a much better understanding now about what kind of flow regimes are needed for platypus and other species. So yes, it's a problem. Whenever you fragment populations, it's not great. You want to have large continuous populations that can interbreed and disperse amongst each other. How much of an impact that actually has on populations really depends on how big the area is up and downstream of those dams. If upstream has a large enough area for a self-sufficient population, then it's not that much of an issue. Yeah, I think the bigger thing is what the dams do to river flows. But again, it's kind of these things, there's synergistic effects. It's like death by 1,000 cuts. They all sort of impact us together. And if we can create dams that enable movement across them through fish ladders, things like that, then it's going to reduce the impact that those dams have. So.
Yeah. And someone has asked, does Emily have a paper out?
There's a couple that have been published, I believe, or yeah, Yeah, it's a good question. The fire one hasn't been submitted yet. We've been sort of working on, I think it's in its sort of final draft at the moment, so hopefully that'll get submitted soon. These things take ages to get published, so hopefully it'll be out early mid next year if it gets accepted. But yeah, really interesting project, and you know, Emily did a lot more detailed analysis on her data, which was fantastic. And yeah, she was good enough to allow us to use some of that data for this project, which was really critical to what we were able to demonstrate.
Reid said coming soon.
Coming soon. Promises, Reid.
Do you expect this research will change the conservation status in New South Wales / nationally?
Look, I don't think in isolation. As I said, I think that data on declines, look, it needs a lot more work, but I think it highlights issues. And I would say that the areas of New South Wales that we looked at are what we kind of considered relatively good habitats. I think the declines in New South Wales would be much more severe in those kind of western drier regions. So that's really raising some concerns. But as I mentioned, we think that the storms have had an impact on both our detection, but also platypus occurrence itself. So I think that needs to get teased out a bit more. I guess what I wanted to do with this, it wasn't part of this grant. I just wanted to sort of use the data a little bit further and highlight some issues. I think the kind of data that we now have over the last 5-6 years from a variety of different projects gives us a really substantial data set to be able to look at that in a lot more detail, and not just in New South Wales, but we've got much more data in Victoria. We've got a little bit of, well, when I say we, collaborators, people we work with, which hopefully will all work together. There's some data in southern Queensland. I think put together, that would be a really amazing data set to be able to quantify these declines and really get a good handle on their true conservation status nationally.
You've answered so many questions today, Josh. We just have one more. So it's, have you used platypus / eDNA monitoring for assessing waterhole condition?
Sorry, I don't quite understand the question.
That's all the info I have.
Sorry, can you read it out again, Jackie?
Have you used platypus and fish eDNA monitoring for assessing waterhole condition?
Oh, okay. Yes, although we don't really have an index. I guess we've certainly done assessments of platypus and fish in waterholes. It doesn't sort of create an index of condition, but it certainly gives you an idea about what's living there and the kind of diversity that's there. So yes, absolutely, either on-stream waterholes or isolated waterholes. Yep, certainly can do.
All right, well, that's all the questions.
Whew. Thanks, everyone. That was great. I love it when there's lots of questions.
So many questions. Thank you so much, Josh, for leading the session. And thanks to everyone else for joining us and participating in the Q&A. Before